to talk a little bit about innovations in understanding brain structure and function. And for that, we're very lucky to have Dr. John Ngai. He's the director of the NIH Brain Initiative at the NIH that involves multiple institutes and many other collaborators. I think everybody on the Zoom today will agree that understanding the basic structure and function of the brain are absolutely critical for advancing cures of all these conditions that we represent that are so devastating. I think you may know that about 100 million people in the US have brain disorders, and that comprises perhaps 1,000 different diseases, some of them juggernauts and very familiar to you, others quite rare, and people are unlucky enough to have their family names assigned to them, and they're few, but they do need help. They need treatments and they need cures. In order to get those, we need to understand what the brain is, what the cells are, what the basic circuits are, how they all work together, and then we can have tangible targets for treatments and cures. And I think that's what Dr. Ngai and his team are getting at. So the ABC has been a very proud advocate of brain research through advancing innovative neurotechnologies, and that's what BRAIN stands for. The initiative was established in 2013, and we've been honored to help them over the last several years. By the way, I didn't want to give short shrift to psychiatric conditions. There are about 400 of those. We all know how critical issues are with anxiety and depression and substance use disorders, particularly since the pandemic. These are problems that can't be ignored. Anyway, we're very thrilled to have Dr. Ngai participating with us today. He has been a very effective director of the NIH Brain Initiative, and I think he's going to update us now on BRAIN's advances over the past year, and then we'll have an opportunity to ask questions about what this all means and a pathway forward together. So Dr. Ngai, please take it away.
Thanks very much, Dr. Rizzo, for that kind introduction, and very special thanks to the American Brain Coalition. The ABC and its partners have been, and its affiliates have been a terrific partner as we seek to accelerate discovery to find new cures. So let's see, are you seeing the correct screen here?
Yes. Great,
terrific, thanks very much. So I just want to give you an update on where we are and to talk about how the Brain Initiative is, we believe, is transforming neuroscience by accelerating the pace of discovery and with the ultimate goal of accelerating the search for cures. Now, I'm going to hazard a guess that everyone here has a family member, friend, or loved one, or themselves are suffering from some neurological disorders like Parkinson's, Alzheimer's, ALS, epilepsy, stroke, or as Dr. Rizzo mentioned, some neuropsychiatric condition like depression, OCD, autism, or substance use disorder. In some cases, these are debilitating disorders. And together, all these diseases, they're exacting a huge toll, both at the personal level as well as on the societal level. And for these individuals, for their families, for ourselves, we really cannot find a cure fast enough. And it's not going to be one cure, it's going to be many, many different cures. So what I hope to convey to you today is that through accelerating the pace of discovery about how the brain works normally and why and how it doesn't work in disease, the resources being developed by the NIH Brain Initiative will help researchers deliver these cures to us sooner rather than later. So let's see, get this thing going here. So I believe most of you here know or somewhat familiar with the Brain Initiative. I won't belabor too much introduction, but just to remind us all, the NIH Brain Initiative, here the goal is to develop and apply new tools for understanding how neural circuits underlie complex behaviors, both in models of health as well as in disease. The Brain Initiative is a trans-NIH initiative. It spans across 10 of the 27 NIH institutes or centers, and it really is uniquely situated for cross-cutting and accelerated discovery in neuroscience that goes beyond the capability of any single NIH institute or center. And we do so by tapping into synergies across multiple fields to address the personal societal challenges imposed by human brain disorders. And in order for us to achieve this goal of better understanding neural circuits and how to apply this knowledge toward finding cures, it's very important for us to leverage emerging technologies to enable these new discoveries about neural circuit function, to use these discoveries as a foundation for new therapies for human brain disorders. And very importantly, our mission ultimately is to disseminate and democratize these riches for the benefit of all. Now, just give a very quick summary of Brain by the numbers. Since its inception, with the first grants being funded in 2014, NIH has invested over $3 billion in over 1,300 brain projects since that initial period in 2014. And with this generous funding from Congress, Brain has been remarkably productive. And our funding right now comes from two main sources: base appropriations shown in dark blue that go to these 10 ICs, as well as 21st Century Cures funding, which was authorized back in 2016, 2017, that adds on top of it. And right now, with these investments, over 5,000 articles have been published to date in top journals of the field across diverse scientific disciplines. So we're really hitting a sweet spot in terms of making groundbreaking discoveries that are building new paths toward understanding and ultimately curing these devastating human brain disorders. And I think we have a shot at doing so within our lifetime. Now, I'd like to leave you today with three takeaways. And because of time constraints, I'm going to have to be very brief, but we're happy to take questions afterwards. The first takeaway is that brain-funded advances in tool and technology development are already making their way into the clinic with a big potential to see positive impacts on human health. So still early days, but I think we're starting to see the payoff already. The second takeaway I'd like to leave you with is that brain-funded teams are developing new resources and technologies that are laying down a foundation for future cures. So this is basically we're hoping that it can advance the timeline for getting at the fundamentals of human diseases and therefore finding actual cures and not just treatments. And then finally, we're changing the way we're doing science, all in the service of the above. We are leveraging talent from different backgrounds and across the globe, actually, to tackle these complex problems and deliver resources that, again, will accelerate research across all domains of neuroscience. And again, this spans not just within any one IC mission, but across all of neuroscience. Okay, so let me just take on this first point. I want to give you one example about how brain-funded investigators are taking best-in-class existing technologies and pushing them further to have an impact in the clinic. So this is just a series of figures from a study that was published late last year by Dr. Samir Sheth's group at Baylor College of Medicine. Now, I'm sure most of not all of you are quite familiar with deep brain stimulation. It's been around for decades, used primarily for the treatment of movement disorders, including the symptoms of Parkinson's disease. And they're kind of pushing the limits more. And if we look at mood disorders, they are highly complex. And DBS was initially proposed and tried out for mood disorders maybe a decade ago. After some initial successes, it kind of hit some hard times because it was found to be ineffective. And part of that probably is because each individual is different. If we look at any particular mood disorder like depression, it's not one disease or disorder. It's probably going to be very, very specific for different individuals. And if you don't target not only the right part of the anatomy, but actually intervene at the right under the right conditions, the chances of success probably will not be so high. So what folks have done using some very sophisticated reasoning, imaging, as well as computational methods, is they have started tailoring these DBS therapies to the individual patient. So what you can see here in the upper left is Dr. Sheth and his team looking at a holographic display of a particular part of a circuit that they believe is going to be involved in driving this particular patient into a depressive state, and one that was until now completely resistant to treatment. And they use this as a basis for where to place the stimulating and recording electrodes. And then from these recording electrodes, they actually monitor this patient for 9 days, and they correlated activity from these different electrode sites with both objective and subjective measures of the patient's mood. And they were able to design a therapy customized for that patient to provide stimulation to move that patient literally from a poor mood state to a good mood state. And it actually pushed this patient into remission from that patient's symptoms after some number of weeks. And there's a complementary studies being performed by other groups around the country, including at UCSF. And this is really, really quite promising, these so-called personalized DBS therapies, adaptive or closed therapies, that literally was the stuff of science fiction maybe 5 or 8 years ago, and now is coming into reality. And currently we are supporting larger clinical trials to see how much more broadly we can apply this and disseminate it and learn not only about how to treat folks with these debilitating diseases, but also learn a bit more about what's driving them. So there'll be a great information to come in the future. So this is just one example of how brain is already starting to show promise in the clinic. And we're very, very hopeful that we can bring some help to folks who otherwise have no hope for any relief. The second thing I'd like to discuss with you just very briefly is that we're laying down a foundation to bring future cures closer to us based on gaining more fundamental knowledge about how neural circuits work. So the basic idea behind this is that in order to understand how the neural circuits in our brains underlie behavior, emotion, cognition, and so forth, we need to have a parts list as well as a wiring diagram for the brain. So we need to know what the circuit looks like, as well as a way to interrogate and modulate these circuits, not only to test hypotheses about how these components interact and how they're wired together, but also ultimately as a foundation for future cures. Now, the big challenge here is one of complexity. So the human brain comprises 86 billion neurons, plus or minus, about twice as many non-neural cells, which altogether form these highly complex circuits and subcircuits. And we need to understand what the parts are and how they communicate with each other to understand what goes wrong in various disease states, including these neurodegenerative diseases that we're also concerned about, neuropsychiatric disorders, as well as things like long COVID, which we believe is going to certainly involve some aspect of neuroinflammation. So again, we need a parts list, we need a wiring diagram, we need a way to harness this information to modulate the circuits with the ultimate goal of intervening in these circuit disorders of the human brain. So brain has started moving very, very intentionally in this area. I'd just like to give you one example of a recent success, a down payment, if you will. And this is coming out of what we call what was just closing out as the Brain Initiative Cell Census Network, where the goal here was to generate a comprehensive parts list of the mammalian brain using very multiple and very sophisticated cutting edge techniques. And this was recently published as a characterization of the primary motor cortex across different mammalian species. It was published in a series of papers in October 2021 issue of Nature, plus another set of papers in Nature sister journals. And it really is a testament to what you can do, what one can do by leveraging large and very highly motivated teams. So we performed an analysis of primary motor cortex in multiple mammalian species at unprecedented depth and breadth, and identified upwards of over 100 different discrete cell types with their characteristics embedded in this information. And this gave us some insights into the organizational principles underlying the diversity of brain cell types. And again, this really showed the power of team science. This is really an example of being able to obtain a resource that can be used as an atlas for everybody in the field. But it really could only have been done with this large consortium. It encompassed over 250 researchers, over 3 continents, from over 45 institutions. And so, is this really going to be a foundation for new cures? Well, it turns out, I think, yes. Already, even though this project is still underway and it's just 5 years old, it's already provided the tools and the know-how that have allowed groups to identify the specific brain cell types that are vulnerable either in Alzheimer's disease or in Parkinson's disease in humans, not in a model system, but in humans. And this provides us critical information for ultimately finding prevention and cures. So it's not just that we've identified cells that are vulnerable. We knew that cells were dying, but we now have a molecular handle on these cells, which allows us now to go in and understand more about them, what genes, proteins, molecules they express, and hopefully how these genes and proteins interact that make them either more or less vulnerable in different forms of the disease. So this is very, very powerful information. I am confident that this will have a big impact in helping us understand much more about how these diseases not only progress, but how they're initially initiated. And this sets the stage for the human brain cell atlas. After all, we are very interested in how the human brain works, and again, the basis for human brain disorders. So to this end, Brain is creating a new way of doing science. I mentioned this as the third point. And based on the success of the cell census project that I just mentioned to you, which is really just kind of a step-up phase, Brain Initiative is now entering into a new phase where it was believed and is decided that this is a great time to invest in a few large projects that really could transform the way that everybody in the field does their work. So the overarching goal of these three so-called Brain 2.0 transformative projects is to change the trajectory of neuroscience research by providing tools and resources that everyone can use, regardless of their problem, their system, to study neural circuits across the brain, and very importantly, to do so at depth and at scale. So the three projects, and they're highly complementary, are the Brain Cell Atlas, which is shifting the emphasis from model species, mouse, toward creating a comprehensive part list of human and non-human brains. Together, so that's a part list. The second project is the whole mammalian brain connectivity project, which is to develop the tools necessary to generate a wiring diagram of mammalian brains. So we have these 86 billion neurons in our brains of these other non-neuronal cells. We need to understand how they connect with each other that actually forms these neural circuits. And then finally, we need tools for gaining precision cell access to these brain cell types that will allow us to both monitor, interrogate, and modulate these molecularly defined cell types and circuits. And together, these three projects, we expect to lay the foundation for future molecular and gene therapies for various neurologic and neuropsychiatric diseases. So this is really where the future is going to be. I strongly believe in every part of my body that with this information, neuroscience researchers will be able to make the discoveries at a much more rapid and a much more deeper clip. So these were the three takeaways I'm hoping to leave you with today. I know I'm running a bit over time. We're already seeing advances in the clinic from brain-funded projects. We're laying down a foundation to bring future cures to us closer. And we're also changing the way of doing science in the service of the above. And there's a fourth takeaway, and that is we are uniquely situated to pursue new and cross-cutting opportunities in technology and resource development that are not and would not otherwise be pursued by any one NIH institute or center. And we're not just adding incrementally to the many great advances being supported by NIH in the neuroscience space, but rather we're proving to be a strong force multiplier that is accelerating the pace of discovery that promises to bring much-needed cures on a shorter timeline. So it's really critical for us to continue building this momentum, to push the field forward, and to make today's science fiction tomorrow's reality. And I'll stop here. I hope I have a few minutes for questions.
Thank you, Dr. Ngai. That was inspirational. And I'm sure that people have many questions and thoughts, and I would encourage them to enter those into the chat. Meanwhile, maybe I will start off with a question. And not seeing anything in the chat as yet, I would ask, with all this opportunity for classification of cells in the brain and evaluating connections that we didn't know about otherwise, there's this great opportunity for precision medicine. I'm wondering how our thinking about inclusiveness and diversity actually plays into the work that you're doing. There are very diverse brains in different groups of people, and some of them are maybe a little bit different developmentally, genetically. How does the work that the Brain Initiative does play into the DEI?
Yeah, that's a great question. Thanks for bringing that up. You're absolutely right. And in fact, every human is different. We're all different from each other in some way, in some subtle ways and some not so subtle ways. I think the experience we're seeing with this DBS therapies for depression is a great example. Just the absolute need to personalize, to tailor the therapy to the human you're actually trying to treat, you're attempting to treat. And that's because, for a couple of reasons, everybody's anatomy is a bit different. The developmental and life history is different, so their circuits are going to be set up in a somewhat different way. And in cases where there are pathologies, of course, that's going to throw in a different set of variables. So certainly that we can see that on the individual basis. Now, in these big projects that we've stood up, for example, the human brain cell atlasing project, we've now just started these projects with an investment of about $100 million a year. And one of the key aspects that we demanded of our funded groups, and these are terrific groups and large consortia, is we have to have diversity in the sample pool. We demanded that there be ancestral diversity, as well as different samples from different points along the lifespan. So if we really are to understand how the human, any part of the human biology works, we have to take into account the variability, the diversity of humans on the planet. And the variability, actually, it's a feature, it's not a bug, right? Human disease mechanisms are going to be common. How they play out phenotypically is going to be subject to these other drivers of variability. And I think that's where the magic, that's where the gold is. That's where we're really going to learn about how organisms normally develop, how they function, and how diseases manifest. So we're really looking forward to really tapping in to the diversity of humans that we hope to study and hope to therefore provide resources for in terms of treatments down the line.
Thank you, Dr. Ngai. I know that Dr. Reznick has a question, but so does Susan Raymond. I'll start with her and then get to you, Mark. Susan's chat is disabled. She is at MTech, and they work on technology development for health issues in the military. And these intersect with civilian concerns. Examples would be trauma, stress, I guess PTSD. And the question is, do you have any involvement in that work?
So yes and no, right? We're not jointly funding projects with the military. And here, there's been a lot of very active involvement in the BRAIN initiative from DARPA. But that we do have mutual interests, for example, in conditions like PTSD and other behavioral disorders. DARPA has quite a few very exciting projects in the neuroscience space. And they by their nature take very much larger risks. The studies that I told you about very briefly about using deep brain stimulation to treat mental health disorders, that the origins of that project can actually be traced back to a DARPA project that started in 2014, right? And it was, when it was proposed, DARPA is very bold. It was proposed. They were interested in dealing with or helping warriors that came back from the theaters abroad with conditions like PTSD. They were looking for ways to help them. So in this case, we certainly have a mutual interest. There are, and again, they can mount projects at different points along the risk spectrum. And that project went on for a while, and they continue now. And now we're starting to see them through NIH-funded projects that are being distributed more widely in the clinic. There are other possible uses of some of these technologies that kind of start veer into dual use, like enhancements, and that's something that we ourselves are not focusing on, but there are probably common interests there. So I hope that answers your question.
Thank you, Dr. Ngai. Now we'll move to Dr. Reznick. Mark?
Hi, thanks, John. That was a lovely presentation.
Thank you.
I should mention that one of the founders of deep brain stimulation for depression was Helen Mayberg, a former board member. Okay, so, in fact, my question relates to that as well. So you talked about the heterogeneity in your initial presentation, and you touched on this, but I'm still a little confused. So we have the heterogeneity for the deep brain stimulation, yet we have a homogeneity for the atlas. Is that gonna happen, or is this going to be a fuzzy atlas?
No, sorry to confuse. So the atlas will be based on a limited set of reference brains, but there will be deep dives across a large number of brains. So what we want to do is we want to understand what are the common features across individuals and what's variable. And the variation is going to come about for a whole host of reasons, but part of it will probably be ancestral, right? But much of it's going to be the life experience of that individual, where they are in their lifespan, their life history, you know, the environment in which they grew up, and so on and so forth. So it's going to be a combination of both. And again, what we need to understand is what's common, what's different. As I'm sure you all can appreciate, some populations, some subpopulations are more or less vulnerable to certain diseases. Well, why is that? Right? I think we stand to learn a lot from the variability if we collect that information in an intentional and forward-thinking way, and are able to kind of connect that information down the line. So we have to recognize that there's no two humans on the planet or alike. Even if we did have the equivalent of an inbred mouse strain in humans, I don't think we'd necessarily want to study it, because, you know, it's not going to be so useful, right? So it's going to be a complex endeavor, but one thing is for sure: what we've learned from whole genome genetic association studies is that if one makes predictions for disease susceptibility based on one population, for example, Europe, people from Northern Europe, those predictions are really great for that population, and they're actually pretty terrible for everybody else. So we're missing a lot. And we're not only missing the opportunity to help more people, but we're missing a lot of biology in there.
Wouldn't it be cool if identical twins were actually slightly different?
Well, I'm sure they're different in the epigenetically, right? And that's one of the things we're looking at as well is epigenetic factors, right?
Thank you. I'm not seeing any other hands right now, but I would ask this. We have an array of organizations that represent all dimensions of neuroscience and neurological disorders, and each of them probably has a particular angle or interest in the Brain Initiative. How can they get involved with the Brain Initiative? Probably one of the things you want to know is what organizations think, because ultimately the support for something like the Brain Initiative and the NIH comes from the public and many different communities in the public. So how can the organizations that we work with interact usefully and fruitfully with your group at the Brain Initiative?
Yeah, so I mean, ABC has already done a terrific job in helping us connect with people that are interested in what we do, and we very much appreciate that. So for those of you here, you're here because you're with ABC. So please, I encourage you. Sorry, Katie, to create more work, but that's one great point of contact. And come find us. braininitiative.nih.gov. Very easy to remember. Even I can remember it. Just go on the website. There's ways to find us. If not, Katie or anybody here at ABC can just connect you with me. We're always happy to talk to folks out there. We need to understand what the needs are, right? I mean, this is, we need to understand what the needs are, what the opportunities are based on those challenges. We're very, very much interested in hearing more from you. So please, we welcome your input. And there are also many other issues that I didn't have time to touch on, which include ethical issues about what we do. We touched on a little bit in terms of perhaps what the military is doing vis-a-vis what we're doing. And for these ethical issues, the issues regarding the ethical, legal, and societal impacts of what we do, it's critical that we have an open dialogue with everybody, right? And you folks here represent many, many different quarters of the patient and research community, and we always welcome the opportunity to have an open and honest dialogue with you.
So, speaking of intervening with our new understanding of the brain, the parts, the wiring diagram, how to integrate it and how to modulate, you gave a wonderful example of precision medicine in a patient with depression. But regarding the military and private citizens, many are injured, in the field, on the road. They may have something like a spinal cord injury. And
Yes.
so one of the things that we need to sort of mediate is the connections that are disrupted. And it seems like the Brain Initiative may have a wonderful substrate for driving research related to recovery of motor function in, say, people who have had a spinal cord injury or a stroke or
Yes.
multiple
Yes.
sclerosis. And I'm sure a lot of organizations will be interested in moving better. So what's going on?
So what's going on in terms of what we're doing? I mean, a lot of some of the technology that we've been supporting is really aimed at after the fact, right? How can we help out folks with these traumatic injuries, stroke, et cetera, live a better life? There's been some really amazing work done with language prostheses, neural prostheses that read out and convert recorded brain activity into either speech or text. But that doesn't give the person back their full life. How do we actually truly restore function? And again, this really comes back to brain's kind of unique perch of providing tools that will allow individual ICs really to pursue the search for these cures in a much more informed and we think more effective way. So we have a few projects we have quite a few projects that are looking at neural circuit function, not only within the brain proper, but also in spinal circuits. It's the Brain Initiative, but we came to the realization that, the brain's connected to other parts of the body. And given that the spinal cord is the CNS, I've had no compunction into really actively pushing ourselves out there. And of course, the function within the local spinal circuits is fascinating, and I think will be the key to helping patients regain recovery. And again, it's a site of possible intervention with different kinds of stimulation technologies, but also this is an area of great interest for regenerative medicine, which is a bit outside of brain's wheelhouse right now. But I would still argue that the more information that researchers have going in, the better studies they will design and the better information they will have coming out. And in fact, it's rare that I can pick up a journal, read a very, very exciting result, whether we fund it or not. There's a good chance that those folks relied on information that Brain Initiative investigators provided, if not the tool themselves. So we're really trying to lay down the foundation that will just accelerate all neuroscience research, really to be the force multiplier here.
Thank you. I mean, this conciliant silo spanning approach is probably very exciting to many of our members. And I think probably to Dr. Kelly, because probably a number of the things you mentioned resonate with the work that he's doing in the FDA. And I know you presented a diagram where you showed all the different institutes you work with. There's so much more to talk about. Katie, I think we're kind of out of time right now. Is that right? So thank you so much, Dr. Ngai. That is just so fascinating and inspiring, and we want to work with you and we want to help. It's obviously that the Brain Initiative is unique and special, that it's continuing to grow, that you have a great vision going forward with microconnectivity, tools for precision access, and a brand new brain cell atlas that can drive new research for treatments and cures. How wonderful. We want to help. Thank you.